Hello everyone, my name is Viola and I will be talking about linking glutamate and GABA genetic variation and gene expression to behavior and brain. And I have no disclosures to declare. I'm going to talk about imaging genetics, specifically linking genes involved in excitation and inhibition to brain structure and behavior. I will walk through an analysis pipeline looking at genetic variation and gene expression and how I relate them to cortical thickness differences and behavioral measures, specifically in autism, since that is what my research is focusing on. But I will also show you how you can use other neuroimaging methods and behavioral measurements to answer your own research questions as well. Because as we know, there are many different neuroimaging methods, and the speakers after me will also get into several of them. And each of these measures on their own might not tell you very much about the underlying mechanisms of the brain and how it functions. But by combining these neuroimaging and behavioral measures with genetics, and specifically selecting genes with certain functions that we are interested in, like excitation and inhibition, we can get a better understanding of the complexity of excitatory and inhibitory balance and its various contributors. But before we do that, I want to give a brief introduction to genetics so that we are all on the same page. Every part of our body is made out of proteins. They are the building blocks of life, and the proteins themselves are made of strings of amino acids. And sequences of these are determined by our DNA, which are sort of the code with the information of what proteins to make and how. Most of the DNA that we humans share are identical. 99.6% of the genes in any person's genome is exactly the same as in any other person. But there are, of course, differences between us, and the 0.4% that is left is what contains the genetic variation between individuals. And within this 0.4%, every person has about 4 to 5 million different variations. And if we zoom in on a DNA strand like we have done here, we see these parts with the letters that you might have seen before. And these letters are referred to as single nucleotide polymorphisms, or SNPs. And the combinations of these SNPs together encode for all the amino acids that make up a whole protein. And that is what a gene is: a bit of DNA that encodes for a certain protein. And the differences in SNPs is the main place where genetic variation occurs, so the majority of that 0.4%. And this is also how genes encode for certain functions. They encode proteins involved in specific processes and functions. So if we want to relate this specifically to the brain, all the cells and their parts, like the receptors and transporters and the neurotransmitters, are made out of proteins. This means that genes encode for proteins involved in excitatory and inhibitory neurotransmission. So understanding genes can then help us understand mechanisms of how the brain develops and functions. And imaging genetics is what we call the methods that we use to try to understand these relationships in greater detail. Every cell in your body contains all of your DNA, or your genome, but different genes are expressed differently in different cells, depending on their function. Specific parts of the DNA, so the relevant genes, are expressed, which encodes for the proteins needed for a certain function that the cell will have. So what is originally one cell in the uterus gets copied, and different genes are turned on in different copies of that original cell, and you get the different cells that make up your whole body. To know what genes are expressed in an individual cell, there are multiple methods to go about it, and one method that I will focus on here is using microarray techniques, which is a fancy method that I'm not going to go into detail about. But here we can see what genes are turned on in what cell, telling us about their function. To do this, you need to take the brain of an individual and look at very thin slices of tissue, extract the gene information from the cells, and analyze that information to read what genes are expressed or activated in the cell. That means looking at the individual cells across the brain to see what genes are expressed where. And as you can imagine, this is a lot of work. And you can only do this on post-mortem donors, or in animals or in petri dishes, since you need to slice the tissue. Thankfully for us, this has already been done in humans, and the Allen Human Brain Atlas has this data freely available from 6 donors, which we will use later. And this was a brief walkthrough of genes, what they are, what they do, and why investigating genes is relevant for understanding the brain. Now we know that genetic variations are variations in the SNPs that are part of our genes, and that gene expression is the process of genes that are expressed or turned on to encode proteins in any individual cell. We also know that genes encode proteins involved in brain functions, since they are part of the development of every protein. And to start using genes to understand excitation and inhibition, we can then select genes that we know encode proteins involved in certain functions that we are interested in. Glutamate and GABA are the most common excitatory and inhibitory neurotransmitters in the brain, respectively. And typically, when we are doing in vivo measurements of excitation and inhibition, these are the ones that we look at. So we can select genes that are involved in the glutamate and GABA pathways in the brain. We selected our genes using Ingenuity pathway analysis, and this is a program that collects information about genes in a canonical way, meaning that they compile information about the functions of genes from literature. And this includes gene expression data, annotation databases, studies looking at up and down regulation and things like that to gather information about which genes are more than chance involved in certain functions. A pathway of genes is a whole pathway of a biological function. Take, for example, the glutamate pathway signaling in the brain. Ingenuity pathway analysis selects all protein encoding genes for this glutamate pathway. And this is, for example, genes involved in the pre- and postsynaptic neurons, which you can also see the outline of here. Glutamate itself is not a protein, right? It's a neurotransmitter. So genes don't encode for glutamate in itself, but we select genes involved in the glutamate pathway. Here we ended up with 72 genes in our glutamate set and 124 in our GABA gene set. And we use the glutamate and GABA pathways here, but there are, of course, many more pathways you can do depending on what biological function or metabolite that you are interested in. And the work I am going to talk about as the example throughout today was published earlier this year, and in it we list all the genes that are included in these gene sets. So if you're interested in looking into this in more detail, I will provide a QR code at the end as well. And here I'm also linking to the website for the Ingenuity pathway analysis here, if you would like more information about them as well. So now we have our sets of glutamate and GABA genes, and we will use these gene sets to investigate genetic variation of glutamate and GABA to behavioral measures and to cortical thickness measures from structural MRI data. And this analysis method is called competitive gene set analysis. And we will also look at expression of these genes, combining gene expression data from the Allen Human Brain Atlas with the cortical thickness measures in our own participants. And this method I refer to as gene expression analysis. So the example that I will use for how we use these analysis methods will use a large cohort of autistic and non-autistic participants, since my research is focusing on understanding excitation and inhibition imbalance in autism. And that is because one of the most popular theories of the underlying mechanisms of autism is that there is an excitation inhibition imbalance in the brain. But we don't really know what kind of imbalance that would be, where in the brain it would occur, or what behaviors that we typically see in autism that this would lead to. And it could be that different kinds of imbalance affecting the brain and leading to different behaviors in different ways that we see in autism. So to investigate these relationships from our participants, we had genome data taken from blood or saliva, brain structure from structural MRI, and behavioral measures capturing the what is typically called the core features of autism. So I will therefore also be mentioning autism a few times during the talk, and there are different opinions and preferences for whether you should say person with autism or autistic person. I tend to go with autistic person because that is a general preference of the autism community. So before I get into the analysis, I just want to briefly introduce the data set as well. So the data comes from the Longitudinal European Autism Project Study, or LEAP, which is part of the AIMS2 Trials Consortium. And this is the largest consortium on autism. And LEAP is, as you can hear in the name, a longitudinal multi-center study across Europe from childhood into adulthood that includes both autistic and non-autistic participants. And these participants are what we call both deeply genotyped and phenotyped, meaning that we have their genetic information, but also EEG and MRI imaging cognitive testing, and a wealth of clinical information in the form of assessments and questionnaires. And in 2 years from now, we will also have data of 3 different time points from them as well. But here we use data from the first time point. So we had data from 638 participants, and here they were between 6 and 30 years old. And we wanted to look at brain structure, specifically cortical thickness, which is a measure of gray matter volume across the cortex, because it's in gray matter that neurons connect and communicate. And differences in cortical thickness has consistently been found in autism, where both increased and decreased cortical thickness has been seen in different brain regions. And this could be affected by glutamate and GABA functioning, since they play a role in the functions that goes on there, and both have been linked to autism previously as well. We measure cortical thickness using MRI, where we make an anatomical scan and then extract the cortical thickness. And we did this here using FreeSurfer, where the images are modeled and parcellated into 34 regions per hemisphere using the Decian-Kiliani atlas. So here we have cortical thickness data for each brain region in every participant. And coincidentally, the Allen Human Brain Atlas data have also already been parcellated into the same FreeSurfer DK atlas as well. And I'll get into that a bit more when we get to the gene expression analysis. Then, to capture the behavioral characteristics of autism, we focused on what are considered core features in our measurements, and these are repetitive behaviors that we measured with the repetitive behavior scale questionnaire, social behaviors from the social responsiveness scale, and sensory processing from the short sensory profile. And these were measured in all of the participants. Then we also used the diagnostic interviews that are used to diagnose autism, and these were measured in our autistic participants. So here we had the interviews ADOS and the ADI. So now we have our structural MRI data, we have our behavioral data, and we have the whole genome data. And with this, I'm then going to use the gene sets for glutamate and GABA as our proxy for excitation and inhibition to link genetic variation to brain and behavioral measures, and then use the Allen Human Brain Atlas to link the gene expression of the glutamate and GABA genes to cortical thickness as well. So now it's time to get into the 2 analysis methods, competitive gene set analysis and then the gene expression analysis. Starting with the competitive gene set analysis, we will use MAGMA. And this is a gene set analysis tool that we use to associate genetic variation of our functional gene sets, so the ones that we selected for glutamate and GABA earlier, with a phenotype of interest. So here that is behavioral measures and regional cortical thickness. What makes this competitive is that it compares the association of aggregated genetic variation in our whole gene set to the association of the rest of the genome with our phenotype of interest. Several kinds of tools exist to do gene set analysis, and they have slightly different ways of doing it, which essentially can lead to testing different hypotheses or having different sensitivities or assumptions. MAGMA has been shown to be more robust against biases like gene size, since a larger gene means more SNPs, which can bias the results. Also to number of participants, number of genes in the gene sets, and account for something called linkage disequilibrium. Linkage disequilibrium means that the SNPs, or the genes that are physically closer to each other, are more likely to be correlated, as in encode for proteins involved in the same functions. Basically, this means that not all combinations of genes are equally likely to occur, and this creates a bias when doing gene analysis, but here we take this into account. The manual that they provide and the publications for how to use MAGMA is quite clear and easy to follow, also for non-geneticists like me, and the tool can be downloaded using the link in this QR code, and it is also a command line interface, so it does not require writing extensive scripts either. Competitive gene set analysis is done in 2 steps. First, we do a gene level analysis associating every gene to the phenotype of interest, and then we get the correlations between the genes, which we then use to account for linkage disequilibrium. And in the second step, we take this output data, so the p-values of the associations from before and the correlation matrix, to associate the whole gene set with the phenotype of interest. And then this is essentially an equivalent of a t-test, where we see if the average association of genes in the gene set is different from the average association of the genes outside the gene set. but there are, of course, a few steps to get there that we're going to go through now. With my own data set, I start out with raw genotype data. That means that we collected our samples from the participants, it was processed, and now what I have available is these 3 file types containing the raw SNPs from all of the participants. So what I need to do first is annotate this raw data, and that is map our SNPs onto genes. To do this, we use reference data. Here we use one that is called 1000 genomes, which is also available on the Magma website. And in the command line, this is then what you enter in just one line. We first call Magma to call the analysis tool itself. And this is how we begin every command line analysis step. Then we add a flag for annotating. We base it on the SNP location that we have in our raw genotype files. And then we reference the 1000 genomes file that we got from their website. And then we name the output files. And out of this, we get a not very interesting looking text file, which we will use in our next step. We also get a log file that tells you the flags that you put in, how many genes they mapped onto each chromosome and things like that. And this is useful to have to be able to reproduce exactly the same steps that you made. It's also easy for sharing what you've done, also since your steps are otherwise just put into the command line. So we have our annotated gene data now and our phenotype of interest. And then what we do next is quantify the degree of association that every gene has with the phenotype and estimate the correlation between the genes. This is done using multiple linear principal components regression that uses f-tests to compute the gene p-values. And this projects the SNP matrix of the genes from the annotation step onto its principal component and then use them as predictors in the linear regression model. In this step, we also fully account for linkage disequilibrium and calculate the correlation matrix between the genes, which we then use in the next step. And this creates grouped associations of the SNPs that were allocated to a gene. So here is the information that you put into the command line. We have the raw genotype data, the annotation file from the previous step, and then we define our phenotype of interest. And here we will use the total score of one of the questionnaires as an example. So let's take sensory processing. And in this step, you can also add covariates. So for our analysis, we included things like age and sex and site. And then when we run this, this is the output. It's a genome-wide list of all the genes that were tested, containing all the resulting p-values from those tests. And in this output file, we first have the list of genes and the p-values for each of these genes, association with the sensory processing in this case. And we also see things like how many SNPs there is for each gene. So we've now aggregated genetic variation onto genes, and we have tested their association with the sensory processing score. And then comparing this step to associating the SNPs themselves, this is a lot fewer tests, which gives us increased power to detect these effects. But then again, in the gene set analysis, in the next step, the unit of analysis is the gene set instead of the individual genes, which will further increase our power. The next thing we do is the actual gene set analysis. When we test the whole gene set that has a functional role, so here the glutamate or the GABA communication pathway, it makes the interpretation of our results easier than when we are investigating individual genes. It also lets us deal with genetic heterogeneity in the sense that you have a polygenic trait, so a trait that does not have a single gene that we know underlie the trait. So for example, that could be sensory processing. There isn't one gene for sensory processing, or more broadly, it could also be autism. So you have multiple genes that are slightly associated with the phenotype of interest. And these genes alone might not have a strong enough association to get over the p-value threshold, but when grouping them together, it does, because it is their common function that is the causal effect. So here that would be the glutamate or GABA signaling pathway having a causal effect on sensory processing. The null hypothesis for the competitive gene set analysis is that the genes in the gene set are not more strongly associated with the phenotype than the genes that are not in the gene set. So this step involves a linear regression model on the gene level data matrix. The residuals are modeled as multivariate normal with correlations set to the gene correlations that we computed during the previous step. and this accounts for the linkage disequilibrium between the genes that are in close proximity to each other. The gene set p-value is the one resulting from a test on the coefficient, testing the null hypothesis against the one-sided alternative. So essentially, this tests whether the mean association with the phenotype of the genes in the gene set is greater than that of the mean of the genes not in the gene set. The command line input here is the result from the previous step, the gene set file and the gene annotation, and then we define the output. This is what our end result looks like. And in our example here with the GABA gene set with the sensory processing scores, we get an above threshold p-value. And we also get an output file containing the stats for the individual genes and a log file if you want to look back at that. And here you can also see the number of participants whose data was included and also the number of genes in the whole test. And when we did this with the glutamate and GABA genes for all of our behavioral measures, and then we also summarized the outputs together. And of course, if you do this many times, we correct our p-values accordingly. We did also do this using the regional cortical thickness data, and that gives us 34 tests per hemisphere per gene set. But for simplicity, let's focus on the gene set associations here with our behavioral measures. So here's a broad summary of what we found. Again, looking at the GABA association with sensory processing, we see that after FDR correction, the p-value is no longer above threshold. We also see that the ADI and ADOS measures, so these were the diagnostic interviews that are mainly capturing other behavioral characteristics in autism, like repetitive and social behaviors, here have associations with the glutamate gene set, which is not the case for the GABA gene set. So what we concluded from this was that sensory processing and repetitive and social behaviors measured in the diagnostic interviews seems to have different excitatory and inhibitory imbalances going on underlying these behaviors. And I also want to note here that these are quite a lot of tests, since one row in each table is one association, but most genetic studies have, thousands of people in their cohorts. Here we have a bit over 600, which is a lot for the kind of cohort that it is, but for genetic analysis, it's not. Although, as mentioned earlier, this analysis method is less sensitive to sample size in terms of number of participants, so it isn't really a concern for power of our results. So I used behavioral measures and cortical thickness here, but it's also possible to include other phenotype measures like functional MRI, MRS, EEG markers is also possible as long as you have a one value per participant variable and it makes sense to connect it to your functional gene set, you can use this tool. Now let's get into the gene expression analysis. As a little refresh, gene expression is the process where certain genes in a cell are turned on to encode proteins for certain functions. And this method was initially developed by Leon French and Thomas Paus and Yash Patel and their colleagues to investigate relationships between cell type gene sets and cortical thickness. They called it virtual histology. Histology meaning studying the structure of tissue, which makes a lot of sense when using cell type gene sets. We adapted this method and instead use our glutamate and GABA gene sets, so therefore I refer to it as gene expression analysis instead. So as mentioned earlier, gene expression data is available from 6 post-mortem donors in the Allen Human Brain Atlas, and this is very extensive data, and the complete data set consists of more than 50,000 measurements of gene expression in 3,702 brain samples from just those 6 donors. So what Leon French and Thomas Paus did was use the fact that the gene expression data is presented as voxels with coordinates in the MNI 152 atlas, and they took this information and mapped it onto the DK atlas that is built into FreeSurfer. They mapped the gene expression data onto the 34 regions per hemisphere, averaging the voxels for each region, giving one value of gene expression per brain region per donor. And they also calculated the median gene expression profile. And they have nicely shared this data for others to use. And that is what we will be working with. If you want to read more about the Allen Human Brain Atlas to FreeSurfer process, this QR code is to the French and Paus 2015 paper, which is great. And there you can also find this data available. They also published a paper in 2018 using this virtual histology method for the first time, where they also share scripts that they use to perform the steps that we're going to go over now as well. What we do here is test whether there is an association between gene expression across brain regions of our glutamate and GABA genes with cortical thickness across brain regions. What I did more specifically was look at cortical thickness differences between the autistic and non-autistic participants. And to do this, we calculate cortical thickness difference profiles in each brain region and gene expression profiles in each brain region. And then we correlate the average cortical thickness difference with the average gene expression. The reasoning here is that if the function of the sets, so the glutamate and GABA pathway signaling, underlies the cortical thickness differences, the mean expression and the cortical thickness difference correlation coefficient should be different for that of a random set of genes. And we test this by creating an empirical null distribution, taking a random set of genes that is the same size as the gene set that we are testing. We repeat this 10,000 times and take that as the null distribution that we test our gene set against. So let's first create the cortical thickness difference profiles. As mentioned here, our cortical thickness data was processed using FreeSurfer as well and was mapped onto the same atlas as the Allen Human Brain Atlas, giving us 34 brain regions per hemisphere. The gene expression data is only available from 2 donors in the right hemisphere, whereas the left hemisphere have data from all 6 donors. So for that reason, this analysis would only be done using left hemisphere data. And since we're interested in understanding differences in autism, we calculated the cortical thickness differences by subtracting the average cortical thickness in each brain region of the non-autistic participants from the average cortical thickness of the autistic participants. And then we have one value per brain region of cortical thickness difference across the whole data set. Now let's create the gene expression profiles. The first thing we do is filter genes that have consistent gene expression so that we can be more confident that the gene expression we include is not biased. And this is done in 2 steps as described in their 2018 paper. So when French and Paus created the DK atlas of the gene expression, they had expression data from each gene for each donor, but they also calculated the median. And with this, they provide the correlations of the expression for each donor to the median. And here we select the genes having a donor to median correlation of rho over 0.446, and this is a Spearman correlation coefficient. Then we use the expression data from another atlas, the brain span atlas, and this has a very wide range of donors. So we select the ones with the same age range as our own cohort, so between 6 and 30 years old, which results in 9 donors. And here we calculate the donor to median expression values in the 11 cortical regions that also overlap to the ones available in the Allen Human Brain Atlas data. And we select the genes that correlate between the atlases higher than R is 0.52. This gave us 2,293 genes. In our glutamate set, this was 29, and in the GABA gene set, we had 42 genes. And this is then the median expression profiles across the brain regions that we then use as our interregional gene expression profiles in the analysis. Now it's time to start linking the gene expression with cortical thickness differences. We calculate the average correlation between gene expression of the genes in a gene set with cortical thickness differences across brain regions. This gives us a distribution of correlation coefficients consisting of the genes in our gene set, and that is the blue parts in this figure. And the average correlation coefficient across the gene set is the dashed lines in this figure. Then we test this for significance against the empirical null distribution that we created, and that is the gray area in the figure. More specifically, this gray area is the confidence interval of 2.5 to 97.5%. And we do a two-tailed significance test to see if the gene set is significantly correlated compared to the null distribution to the cortical thickness differences. And that tells us whether it differs from a random set of genes in the genome. And this is then indicated visually in this figure with the dashed line of the gene set average correlation laying outside of the null distribution in the gray area. So let's take a look at the stats and the results. When we first ran this across our entire data set, we saw no significant correlations. However, our participants were between 6 and 30 years old, right? And we know that cortical thickness changed a lot during development. So we also divided them into age groups of children, adolescents, and adults. And then we got significant and different results. In children, we saw no significant associations, but in adolescents and in adults there were, but in opposite directions. In adolescents, this was positive, and in adults, this was negative, which you can see in the figures here where the dashed line with the average correlation coefficient are on opposite sides of the null distribution in the age groups. So this shows that increased expression of glutamate and GABA genes are significantly correlated with larger differences in cortical thickness between autistic and non-autistic people. And to make a bit more sense of this, we plotted the cortical thickness differences and the gene expression profiles across the brain regions. So on the top row here, you see the cortical thickness differences in the 34 left hemisphere regions of adolescents and in adults. And in the middle and the lower rows, you see the gene expression of the most strongly correlated gene in each gene set. Here, the red colors are indicating a positive cortical thickness difference or gene expression, and blue colors are indicating a negative cortical thickness difference or gene expression. And you see in adolescents, the gene expression is in quite a similar direction compared to the cortical thickness differences across brain regions. Whereas if you look at the adults, the gene expression is more in an opposite direction compared to the cortical thickness differences. So again, we show here that increased expression of both glutamate and GABA are associated with larger differences in cortical thickness between autistic and non-autistic people in adolescents and in adults. So if we are to put our results together and make sense of them, we saw from the competitive gene set analysis that associations between glutamate and GABA genetic variations relate differently to autism behaviors, suggesting that balances between excitation and inhibition are playing different roles in different behaviors linked to autism. And in the gene expression analysis, we saw that increased expression of these genes also relates to larger differences in cortical thickness in autistic people as well, but that these effects are different throughout development. So together, this shows that both glutamate and GABA genes play important but complicated roles in autism, linking to behaviors in different ways, but also to structural differences during development. So the overall conclusion here is that excitatory and inhibitory imbalance is present in different ways, affecting different aspects of autism behaviors and brain development. So with this, I've shown you using my own analysis pipeline how we can use these imaging genetics methods using cortical thickness data and behavioral measures, but these tools can also be used with other measures. So for example, with MAGMA, we could use different neuroimaging measures, like I mentioned earlier, like EEG markers that make sense to relate to excitation and inhibition, or other MR modalities that might be interesting, as long as you have a biologically relevant reasoning behind your research question. A lot is possible to do here. Also with the gene expression analysis, you could use it for other group comparisons like I did here, or divide participants based on other characteristics and compare the results. For example, to build on previous findings within the consortium with this data set, we also divided our participants into sensory processing subgroups, but there we found no differences in the cortical thickness to gene correlations between the groups. But we can, of course, also select different sets of genes that relate to excitation and inhibition as well. There are many possibilities for using imaging genetics to investigate excitation and inhibition in more detail in vivo in humans. There are, of course, also limitations to what you can do and how to interpret your results as well. So, for example, we are not able to make any conclusions regarding actual concentrations of glutamate or GABA in the brain here. Genes can encode for both up and down regulation, and here we looked at whole gene sets, not dividing them by functions of certain genes or gene variants, for example. But what we can do is find relationships between modalities that are not typically looked at together in humans, and use this understanding to guide future research questions and methods to increase our understanding of the links between excitation and inhibition to brain and behavior. And this includes the measures that I used here, but we can also use things like MR spectroscopy, looking at metabolite concentrations, EEG markers, other behavioral measures, or pharmacological studies. We can also use longitudinal data to look at changes over time or through development of the measures that we are interested in. So, for example, we are also planning on going to do this when we have the longitudinal data from this sleep cohort, for example. There really are a lot of possibilities here. But with this, I want to end here, and I hope I could make these methods seem a bit less overwhelming, also for those who are not familiar with genetics from before. And this work was also published earlier this year, and here is a QR code with a link to that paper if you want to read more. And here you can also find the lists of genes that we included in our glutamate and GABA gene sets. And I want to thank everyone working on this with me, not just the people that are named here, but everyone working on the consortium, and especially also thank you to all of the participants. And thank you so much for your attention. And if you would like to reach me online, here is a little bit of contact information. Thank you.